What's up, guys? Alexander with Date Psychology here. Today I want to share with you an older study in the neuroscience of facial attractiveness. This is a study from 1998 using PET scan, positive emission tomography. A little bit of background here, and I'm sure that everyone who has taken an introductory psychology course, cognitive psychology or whatever, has seen this case, a man named Phineas Gage. He was working on a railroad. He put some explosives into whatever it was designed to create the hole, and it launched this tube, this spike up through his prefrontal cortex. And following that, he had major changes in his personality. He became a totally different person after that fact. And reflecting on this case, one of the early examples in cognitive psychology, in neuroscience, that linked regions in the brain, in the prefrontal cortex, with behavior and personality, that sort of a thing. So this was kind of the background going into this that the authors of this paper mentioned. Does it really relate closely to this? No, but just to give you an idea. A little bit more background here. We're looking at PET scans. What is that? Positron emission tomography, right? So this is brain imaging that uses a scanner with an injection of a radioactive isotope, right? From tomos, meaning slice, and graphy, meaning to write or to create an image of, in that case. So why slice, right? Because the way this brain imaging works is it's taking pictures in little slices across the brain, this dimension as well, this dimension as well. And from those different angles, it's able to create a 3D image or a representation of the brain, the same way that MRIs now, magnetic resonance imaging, is used to create this kind of imaging of the brain. So PET scans have been around for a little while, as you can kind of guess from the date on this, 1998. And what they're really used for to this day is measuring brain metabolism, right? So when you have, for example, lesions in the brain, when you have drug-induced changes in the brain, they're kind of good for that. They give you these 3D images of the brain. And what they're specifically used to measure, particularly in the case of this study, is the regional cerebral blood flow, right? That's a measure of brain metabolism, so, and neural activity in that case. So how does that work exactly? The idea, the concept behind this, the principle, is that when you use a region in the brain, when there is a region in the brain associated with perception, behavior, any kind of activity, it requires more blood flow, right, to work, basically. Similar to how if you move a muscle, then blood comes in, right? So it's through measuring that flow in blood that you can see corresponding parts in the brain that are associated with perception, behavior, activity. And in this case, what we're looking at is the perception of faces, specifically attractive faces, unattractive faces. Very clear study design in this, I think, especially for a study in neuroscience, which can get very, very complex. There's one experimental condition. Subjects are shown pictures of faces, a long series of pictures of faces. These are all female faces, by the way. They either rate these faces as attractive, as neutral faces, or unattractive, right? The respondent decides. So this is something that is not decided ahead of time, like we see in a lot of research on facial attractiveness. First, faces are picked for being attractive or unattractive, and then we see how people respond. In this case, the rater of the faces is essentially the same person that is undergoing the experiment. Then we have two control conditions. So what are these? These are the responses that the first experimental condition is going to be compared to in the statistical analysis. In one of these, they're looking at faces again. In fact, they're looking kind of at faces in both. But in the first one, they're looking at faces, and these are going to be facial emotions. It's going to be an emotion recognition task. So they're shown faces that are angry, happy, sad, and they're asked, identify that emotion. And then boom, they have to identify. That's an angry face. That's a sad face. This is putting the participants' attention on faces specifically. In the other control condition, it's a background color identification task. So these faces are superimposed on a background of different colors. And then the participant is asked, identify the color. So they're not looking at the faces, but they can still perceive the face at the same time. So that's important because there may be brain regions, right, that, and there are brain regions that are involved in the recognition of faces when you have attention on those, right, and brain regions implicated in attention at all, as opposed to just perceiving something but not having your attention focused on it. So here we have a condition where the participants are still able to see the face, but they're not directing their attention at that face. Ideally, their attention is being directed elsewhere, at the background, at the color that they're supposed to identify. So two different conditions that are being compared against the identification of attractive or unattractive faces. We're looking at cerebral blood flow, right? So the way that is done is after the image of the face is created, you have a voxel by voxel comparison. Think about in a 2D image, right? You have pixels and it's every little box, tiny little boxes that are made to create the image. In a 3D image, it's a voxel, right? So it's the same thing, but it's 3D, it's called a voxel. And it's by comparing those voxels statistically across conditions that you're able to create an image of where some have more activation than others, as well as to perform the statistical tests that show you was there actually a difference between areas activated associated with these voxels, and also is that difference meaningful? Is it statistically significant? How large is it? And so forth. So what are we looking for then? What is the hypothesis here? Ideally, more activation or less activation or some kind of different activation in regions of the brain when exposed or identifying these faces that are attractive or unattractive. So, as I mentioned, colors, emotions, attractive faces, why all of these? Why don't they just compare something completely different, right? Well, you have to have faces in both conditions, right? You compare it to something else like viewing a banana. We have specific regions in our brain associated with facial recognition. Very, very important, right? You can have lesions in your brain in certain areas that impact your ability to recognize faces at all following brain trauma. Some people can't recognize the faces of family members. They can't remember new faces and so on. But at the same time, you also kind of have to distract people from, as I mentioned, that attention to faces. So a good design here that they accounted for that. They created that color background image. I won't go any more into that because I've already explained it. Right into the results here. We see activation in response to faces when being rated as attractive or unattractive in the anterior frontal cortex. We also see it in the orbital frontal cortex. Here is a diagram of the brain with numbers that indicate the region. I've put the lines up here that kind of show you where they go to. Now, these are brain regions that were activated differentially in the attractiveness condition compared with both the color condition and the emotion recognition task. So if we had gone more into detail into this, which I decided not to do to keep this video very, very short, you will see that some brain regions are activated differentially in the emotion versus the attractiveness task, and some are not activated or activated differentially in comparing the tasks between color recognition. So these are the ones that are different from both, right? Why is this the main result? Why is that more important? Because this is excluding the effect of the other two conditions, simply recognizing a face, or an emotional face for that matter, recognizing emotions, as well as recognizing a face without attention on it, and the additional recognition of a color task. So the idea is to isolate areas of the brain that are associated specifically with a recognition of facial attractiveness. Additionally, we also see activation in the insular cortex. Something, if we look at the last slide, same in this slide, you'll see at the top it says left hemisphere area. Some of these regions had bilateral activation. Bilateral meaning both sides of the brain, right? The brain is a bilateral structure. Typically, it looks pretty similar on both sides, right? It's a mirror image of itself. But we don't always see bilateral activation. In some of the past tasks, bilateral activation was actually seen in comparison with the others. For example, there was bilateral activation in the anterior frontal cortex when compared with the color task. So what does that mean? Probably that we're seeing activation both due to the attractiveness, and we are seeing activation both due to the recognition of a face. So when you cut that out, when you control for that, all of the activation that we see in this study related to the attractiveness conditions are all in the left hemisphere. So what we see here is unilateral activation that may be unique to facial attractiveness, while facial recognition may be more bilateral in that case. Here we see activation in the insular cortex as well. The insular cortex, I picked a different image, you can see peeled back, right? It's on the inside, the insula. So it's not something that you're going to be able to see on the outside of the brain. It's a deep structure within the brain. You might be wondering, like, what do these brain regions do? We can go into that as well. But knowing this, I'll just preface this and I'll get into this in a little bit in a minute. Don't try to link what these areas of the brain do apart from facial recognition too closely with facial recognition itself. Nonetheless, let's just a very brief overview of some big things that are associated. So in the anterior prefrontal, information processing, right? A lot of what we're looking at here in the cortex at all is going to be related to executive function, consciousness, that sort of thing, decision making. So information processing, this is a hub that coordinates information from other regions of the brain. That's something I'll go into a little bit more that's actually pretty important. Executive function, problem solving, decision making, and focus and attention, right? So knowing that it has a role in attention is important, since we have these two conditions as well, that the color versus the two facial tasks that would, in other words, account for direct attention on the face. In the orbital frontal cortex, again, similar, very similar what these do. And the reason for, well, maybe I'll explain that in a minute. Decision making, okay, emotion and reward response, and that's particularly reward response in response to making a decision, right? You make a decision, you get a reward of some kind, or a punishment for that matter. Activation, so inhibition. Inhibition is, how should I explain this? When you try to suppress a thought, when you try to suppress an emotion or an action for that matter, right? So that you can kind of get your mind in order, that you can focus attention on things. Inhibition has an important role in that. And also the processing of values, right? Of meaning, what is important. And the insular cortex. Okay, so the insular cortex actually has a role in a lot of autonomic behaviors, right? Things you don't control. But as far as things that you may experience more as conscious processes, sensory processing, it has to do with your awareness of your own body in space. You know, like, I know my hand is over here. Oh, well, thank you, insular cortex. I know that because of you. You told me it's over there. You know, I know that if I move forward too much, I'm going to run into something. Those are kind of important roles that we have associated with the insular cortex. Emotions, social behavior as well, and emotions specifically related to social behavior, right? What social behavior is rewarding and what isn't. Important roles of the insular cortex. There are many, many more things that these brain regions are associated with that are not listed here. Many, many, many more. Can't go into all of it, but these are a few of the big ones that are kind of classically associated with these regions. So, as I said, I was going to mention something, so here's that part. There's a few caveats to consider. The brain isn't modular, and this is kind of the way that we typically think of the brain as being modular. It's kind of the way that the brain is explained. You'll see a picture of the brain with different areas highlighted. You know, this is in red, this is in green. I'll say this is the thinking spot of the brain. This is the memory spot of the brain. Okay, a much more modern and current model of the brain, understanding of the brain, is thinking of the brain in terms of brain connectivity. Certainly we see more activation in certain regions associated with certain processes, right? Something, for example, like the hippocampus being closely associated with memory. When we do memory tasks, we see a great deal of activation in the hippocampus. At the same time, when we do memory tasks, we see a great deal of activation all over the brain in strange little spots, right? And we see a lot of connectivity going back and forth. And it's much better to kind of understand the brain as being a network of connected nodes, so to speak, rather than being modular. Like, oh, this is the area associated with this, this is the area associated with this. So importantly, you know, following this video, don't go home thinking, for example, like, oh, these are the brain areas of facial attractiveness. There are many, many more studies on facial attractiveness out there that associate it with other areas in the brain as well. This is a very early one, right? This is from 1998. So these were some of the first ones that were kind of identified, which is why I wanted to share this study with you. But there's much, much more going on there. Another important thing to consider, I said I would mention. I listed some things those areas of the brain are associated with. A lot of people make a mistake of thinking that you take brain imaging and you can look at where the brain is activated and then assume consciousness, that you can assume feelings, emotional states, experiences, like you can see activation in the prefrontal cortex and think, I know that person is thinking really hard because he has activation in the prefrontal cortex. No, it doesn't work that way. You can do a cognitive task and you can associate that with activation, but you can't take the activation and turn around and think, they're probably doing that cognitive task. It doesn't work backwards that way. So a big mistake a lot of people make when interpreting neuroimaging results, but that's the case. And what that also means is that we can't link facial attractiveness with any of the other features, or I should say roles, that were listed in these brain regions, right? In the insula, in the orbital frontal cortex, and that sort of a thing. Just because that area has some kind of role or has activation in response to, say, an emotional decision-making task and a pretty face, doesn't mean that like emotional decision-making and pretty faces are actually cognitively linked in any way. They could be, that's one starting point. You could go from there and you could say, I see activation in response to these two things. Are they linked? And perhaps you could design future research to kind of test that hypothesis. But from that activation alone, it's not a lot to go on. So be very, very cautious when you see like, these areas in the brain are associated with emotions, facial attractiveness. So we have this link between facial attractiveness and emotion. Maybe we do. It certainly isn't implausible that there's a link between, attractive faces and emotion. Certainly, even from, a subjective point of view, that seems accurate. But it's not something that you really want to infer from the associated regions of the brain being activated in these two different conditions. Important in these results, okay, so I mentioned brain activation in response to the attractiveness condition, as well as the conditions that were the color and the emotional faces condition. Activation in response to the attractiveness condition was different when viewing unattractive versus attractive faces, right? So the left anterior frontal cortex was associated more with the unattractive faces, but it was that left frontotemporal junction that we actually see more activation when associated with attractive faces, right? When viewing attractive faces. And that's a little bit closer if you look, both as far as its actual location as well as its connectivity, right? Because brain connectivity sometimes follows location. Sometimes two areas that are close, good connectivity between the two, and sometimes that's not the case. Sometimes you see connections from way over here in the brain and it's way over here, and the network is not temporally located. The regions aren't located close to each other in that case. But in this case, they are located kind of close. And, so that's the orbital frontal cortex, which is the area of the brain. It's called that because it's right behind the orbits, right? Right behind the eyes and the insula. Again, remember, peeled back a little bit deeper in there. So the take-home point here, guys, different areas of the brain were activated differentially when exposed to these attractive versus unattractive faces. Mentioned before, we'll go over this again because it's kind of important, probably the take-home point here, is that the activation from attractive faces is not the same as the activation from simply viewing a face, right? You see, because that's the first question really that I think anyone would ask, right? They would say, okay, so we're viewing faces. They're attractive or unattractive. Do we really know that this is not just brain response from viewing faces? So, well, we see the differential activation in unattractive and attractive faces. So already we know that there's some kind of difference there depending on the face, specifically related to attractiveness. But again, that's what the color condition, where they're not directly viewing the face, where they're processing the face, because certainly it is in the visual field, right? It's being taken in by sensory perception, but the attention is not being focused on. The attention is being focused on the background. So this design kind of allowed for that control for passive versus active facial activation. And another thing to consider, as I mentioned, it's kind of a subjective experience that attractive faces are arousing, not even sexually arousing. I mean, emotionally arousing, that they create emotions in people. So showing faces that had different emotional expressions, I think, was also a good way to kind of tease out perhaps the link between, we're looking at attractive faces, but are we really seeing a unique response to attractiveness, or are we seeing a response that is actually more closely associated with emotions related to attractive faces? So perhaps that recognition of emotional faces was also a good step that these researchers did in their control here. So a couple of limitations from this paper we should mention, just so we talk more about neuroscience subjects worth going forward into this. Very common, very, very common to neuroscience, especially to neuroimaging studies, right? Studies that use machines to take pictures and images of the brain. Very small sample size here, guys. 6 people. You think that's very small. The median sample size, you can follow the paper linked here in the slide, in neuroimaging studies, up to this date, this is an old study, but this is still something that's going on, is 12 people. So what are the issues and the limitations of having such a small sample? The biggest one for me, right, is going to be your ability to detect an effect. Unless you have a very large effect, a real effect, if you have a very small sample, you might miss it, right? So you might get the kind of error where you reject, or I should say where you can't reject the null hypothesis when the effect is actually real. How do you detect smaller effects? How do you detect effects that are small but real? Probably the best thing that you can do is increase your sample size. Hard to do in these neuroimaging studies. Why? Because they're time consuming to do. Running the machine is expensive. I was just told in a class the other day, it cost about $100 to put a person into an fMR machine today, but in the 90s it cost about $6,000 per person. Is that the same as one of these PET machines? I imagine it's pretty close because the machinery is not that different as far as, the space that it takes up at the very least. So another limitation, of course, of these very, very small sample sizes is generalizability. You get down to something like 15 or 30 people, and you can't even know if the central limit theorem holds, assuming that, it was even supposed to be generalizable to the general population anyway. This concerns me a little bit less, and the reason is because the vast majority of research, these neuroimaging studies, they're not generalizable samples anyway, right? The biggest key to generalizability is, your sample has to be taken from the population that you want it to be taken from. So if your samples are all student samples, kids taken from the university, you can't really generalize to the whole population from those, regardless of your sample size. Doesn't matter if it's 30 or 30,000. Your sample will only ever be representative of the population that it is taken from, and even then it will only be representative of that population if it's truly a representative sample, which we approximate by making it a random sample. So most research in psychology and neuroscience, not really, strictly speaking, representative anyway. Does that mean that it's bad or that it's truly not generalizable is kind of another question, perhaps more in the philosophy of science, because assuming, that brains work similarly to some extent, even if you take a sample that is not entirely generalizable, the findings of that will probably apply to individuals, groups, or something like that outside of the population that you sampled. But can you assume that, strictly speaking, from a formal point of view? Certainly you cannot. So something to consider. Another limitation of this that I noticed that I thought was perhaps a limitation. The participants are the ones rating the faces as attractive, right? So these are faces, presumably, that the participants said, I find that face attractive. Now, a lot of research in facial attractiveness does this, and at the same time, a lot of research in facial attractiveness is looking for faces that are more objectively attractive. And the way that you would do that is you would have those faces be rated by a group of raters. It could just be another sample. It could be, other researchers. It doesn't matter that much because actually inter-rater correlations of facial attractiveness are kind of high. They're usually over 0.5. So people tend to agree more or less on what an attractive face is and what an attractive face is not. Having people rate the faces that they find attractive could be a confound, right? It could be a confound. It might be something to consider in other studies. And like I said, there's more research in neuroscience on attractive faces that has been done, and I'm sure that some of it has involved faces that have been rated ahead of time. This isn't something that's wrong, what they've done here, but it's just something to consider that these are faces that they subjectively found attractive. They could be different from faces that were rated as attractive in a more objective sense, faces that would have had that convergent, I guess, a composite rating across multiple judges, so to speak. Would it be a better result? Perhaps not. Would it be a different result? It could. It could be a different result. So something to consider there. Final thoughts here. And with this, I'm just going to ask you guys one question. Respond in the comments. Let me know what you think about this. So certainly there's a lot of talk when people discuss facial attractiveness, right? And not just facial attractiveness, but desirability in a relationship. When people talk about love, about who is attractive and who isn't, in any sense. And the idea is, of course, that beauty is in the eye of the beholder. It's almost sort of a narrative that beauty is entirely subjective. Now, I've talked about this in past videos. There seems to be a large subjective component to beauty, but also at the same time a large shared or objective component to beauty. So do you guys think that when we see differential activation in the brain, right, in response to attractive faces over here, we see unattractive faces over here in a different brain region, do you guys think that indicates perhaps a more objective basis for beauty, that beauty is located in the brain? Let me know what you guys think, guys. Like the video, subscribe, leave a comment, and I will make another one for you guys very soon.